Good morning, dear students. I am Dr. Anmol Kumar. Today I will take the topic of microtomy and different types of microtome of the unit first. In my last video, I have discussed about the tissue processing steps. In that steps, you have processed the tissue and finally you embedded the tissue into the paraffin wax for the block preparation, and finally you made the block, and that block is ready for the sectioning purpose. So now we have to section the tissue block for obtaining the thin paraffin sections. So for getting the paraffin section, you need to have some instrument. That instrument name is microtome. So microtome is a precision instrument which is used to cut the thin section of tissue, and that tissue is finally used for the microscopic study. So in this microtome, you can get the section of various thickness from 50 nanometer to 100 micron. But in most of the case, you should have just 4 to 5 micron is enough for your all type of diagnostic studies. So this microtome is divided into various types depending upon the types of tissue, your requirement and your mechanism of action. For whatever microtome you are using, but those microtome having the three main part. First part is the blade holders and blades itself. Next part is the sample holders, and third one is the main body of the microtome, which provide the mechanical device to the sample holders to bring the samples towards the blades and to get the sections. So in the next few slides, I will discuss about the various types of microtome which is being used in the various type of histopathological laboratory. So first of all, I will discuss the rotary microtome. This is the most common types of microtome which are being used in the several types of laboratory as well as in research application labs. So this microtome is also called as a minute microtome because it was discovered by the scientist Professor Minot. That's why its name is minute microtome. So mechanism of this microtome is based on the rotatory action of the wheel. So if you rotate the hand wheel, then the sample holders will move vertically towards the fixed blade and finally section will come out, right? And there are three types of microtomes are there in case of rotary microtome. First one is the manual one, second one is the semi-automated, and third one is the fully automated. In manual one, actually there are two types of wheels are there. One is coarse wheel, another one is the hand wheel. In hand wheel, what will happen? The sample will move vertically, while in case of coarse wheel, you can set the position of sample holders. So in case of manual, both the wheel we have to handle manually, right? But in case of semi-automated, any of the wheel, either you can say the hand wheel or the another wheel, coarse wheel, can be automated, one of them. While in case of fully automated, both the wheels are computerized control. Just you have to set all the parameters and automatic they will cut the sections. But in all the labs, mostly they will use the manual rotary microtome. I want to show one video of a manual rotary microtome, how it's work. Then you will get the better idea of the function of this rotary microtome. See, here you can vertically move the sample holder using the hand wheels, right? See how the sample is moving vertically toward the knife. And another wheel is there, which is back side of the microtome. This is called the coarse wheel. Through this wheel, you can set the position of the sample holders. And this is the knob to set the section thickness. And another one is the blades. You can put the blades in the sample blade holders, and where you can get the sections, and finally you fix the blade here, and finally this thing you will get the section using this microtome. So there are various advantages of the rotary microtome. That's why this microtome is very useful in all the labs. First of all, you can get the section of various range like 0.5 micron to 60 micron, and this type of rotary microtome is useful for all types of tissue, whether it is a fragile tissue, whether it is a hard tissue or it is a small tissue. So this is very user friendly also. And another advantage of this microtome is that it is very stable. If it is very stable and heavy, you will get the very good quality of the section, and it is a flat section, right? And in this case you will get the serial section. In this, what does it mean? All the sections are attached to their edge. So due to lot of advantage of this rotary microtome, this microtome is being used in several laboratories. Now I will discuss another microtome. This is called the rocking microtome. This microtome is oldest in design, and this microtome is also called a Cambridge rocking microtome because it was discovered in the Cambridge company. So this microtome is very cheap and reliable, and it's require very low maintenance, right? The mechanism of this microtome is based on the rocking movement of the cross arm. One side of the cross arm is attached with the micrometer thread, and other side is attached with the sample holders. Now this thread is wrapped over the wheel, and through this wheel one operation lever was attached. Now when we are pushing the operation lever, then wheel is rotating, right? When wheel is rotate, then it will stretch the thread, and finally it gives the rocking movement to the cross arm. Now this cross arm attached with the sample holders, which holds the tissue block, it will strike to the knife, and it will give the thin section. So this is the mechanism of the rocking microtome, and this microtome mostly used for the paraffin section, and it will give the section of around thickness 1 to 20 micron. Now in this video you can get the better idea how this microtome is working actually. See, this microtome, this is the cross arm and this is the thread. When we are pushing the operation lever, it is giving the movement to the cross arm, and the sample holders which contain the tissue block, it strike to the knife and give the thin section. So now this I will discuss another microtome. This is called the sliding microtome. This microtome is a dangerous type of microtome because here we are using the exposed knife, so there is always a chance of injury. So while handling this type of microtome, we should be very careful. Now in this microtome, what will happen? The knife is sliding over the top of the specimen. Now it will give the thin section, and this type of microtome is mostly used for the celloidine embedded tissue, right? And this celloidine is another type of embedding medium which is being used to embed the tissue which having the hollow space, like the brain tissue, eyeball. This type of tissue is embedded by the celloidine. So this type of tissue embedded block which sectioned by the sliding microtome. You can see this video where you will get the better idea of this. So now this knife is sliding over the tissue block, right? And it gives the section. This is another microtome. This is the saw microtome. This saw microtome is used for the cutting of the undecalcified bones and tissue and teeth. In this case, there are a saw which is rotating very high speed at the 600 RPMs, and you have to move the tissue towards the blade, and you will get the thin section in. So in this case, very thin section is not possible, so you will get the section only more than 20 micron. And moreover, in this case, always you have to pour water onto the blade, so it will not generate too much temperature, otherwise it may destroy the samples as well as the blade. So I want to show one video where you will get the better idea of this microtome. So in this case, she is taking the bones toward the blades, and finally she will get the section of the tissue in this using this microtome. And you have to move the bones very slowly, so you will get the good sections, as well as the water should be always poured onto the blades and the tissue material. Now this is the another microtome. This is called the ultra microtome. This ultra microtome being used for the very ultra thin section to study the ultra structural feature of the cells. And this microtome basics is similar to the rotatory microtome, but this microtome is very highly sophisticated and precise, so that you will get the section of very small thickness in the nanometer range. And the blades in this case is coated with the diamond or glass materials or sapphire. So I want to show a video picture here. See, this is a picture of the ultra microtome, and you have to bring the these sample holders towards the blade under the microscopic control. And after the sectioning, the tissue section will float onto the water, which have to be taken carefully onto the TEM grid for the microscopic study. Now this is another type of microtome. This is the cryostat microtome. This cryostat microtome is just like the refrigerated cabinet in which the rotatory microtome was housed and you adapted to cut the frozen tissue. And while you know the tissue contains some water, so the water become ice at its freezing point. So the ice itself working as a embedding medium and allow for sectioning purpose. So here no need to freezing fix the tissue, directly just frozen samples can be used for the sectioning purpose, right? And in this case what will happen, this you should, all the cabinet is provided with all the temperature control, fan and everything. So temperature will maintain around -10 to -40 degree Celsius inside the cabinet, so which will freeze the tissue inside the cabinet itself. And this type of microtome being used for the immunohistochemistry study, because in IHC the antigen should be maintained. If you are using the tissue and process through the normal tissue processing, the antigen will lose, but if you freeze the sample directly at the low temperature, the antigen will maintain and you will get the results in case of IHC. And this microtome also used in the rapid histopathological diagnosis in the operation. And another application of this microtome in the demonstration of labile enzymes, lipid and brain structure. So this is the images of various example of cryostat microtome where the refrigerated cabinets are there and they used to cut the very frozen samples. Here I summarize the all types of microtome for a quick review. Hope you understand all the functioning of the microtome. In the next video lecture I will discuss about the process of the staining process and how to get the sections. Thank you.